Hello, everyone, and welcome to today's webinar in the SERDP and ESTCP webinar series. My name is Rula Deeb. I'm a principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I will be facilitating today's call. The webinar will consist of a brief overview of SERDP and ESTCP by Dr. Andrea Leeson, followed by a list of upcoming webinars in the webinar series. After Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features two presentations on the assessment and treatment of contaminated sediments. First, Dr. Todd Bridges will talk about the roles of biology, chemistry, and exposure in the development of resilient remedies. He'll be followed by a brief Q&A session. Then Dr. Kevin Sowers will discuss the in-situ treatment of PCB impacted sediments by microbial bioaugmentation. We will conclude with a brief Q&A session at the end of the webinar. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions in advance of the Q&A session. On the slide, we have provided a few suggestions in the event that you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line. However, if you continue to have problems, please submit a comment using the chat box in the left corner of your screen. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program Area. Andrea has been with SERDP and ESTCP since 2001, and before that, she was a research scientist at Battelle Memorial Institute, where she conducted research on in-situ bioremediation. And with that, I turn that over to you, Andrea.
Thank you, Rula, and I'm happy to welcome everyone to SERDP and ESTCP's webinar today. SERDP is the Strategic Environmental Research and Development Program and was established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. We fund both fundamental research as well as advanced technologies development that ultimately is meant to impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under SERDP or other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs, with much of SERDP research occurring at the lab and pilot scale, with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting lab efforts are conducted. There are 4 program areas in SERDP, 5 in ESTCP. The Energy and Water Program area is only in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Climate Change, and Weapons System and Platforms, are SERDP and ESTCP programs that are managed jointly by a designated program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Environmental Restoration Program area. Environmental restoration has essentially 5 main areas of research: contaminated groundwater, contaminants on DoD testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. Our webinar series will be highlighting research and demonstration efforts from each of the 5 program areas. As you can see, our webinars will cover a broad range of topics, with upcoming topics covering munitions response to emerging contaminants, to watershed and stormwater management. The next webinar, based on research and demonstrations that were conducted under the Environmental Restoration Program area, will be on December 3rd and will focus on the issue of emerging contaminants. You can find more information about our upcoming webinars on the SERDP and ESTCP website, and you can find the archived webinar series at that website also. Registration is now live for the webinars on munitions response as well as on emerging contaminants. Thank you, and I hope you enjoy the webinar today.
Thank you, Andrea. And it is now my pleasure to introduce Dr. Todd Bridges, who is the U.S. Army Senior Research Scientist for Environmental Science. Todd's primary areas of research activity focus on the science and engineering of sustainable infrastructure development, the development of risk and decision analysis methods applied to water resources infrastructure and environmental systems, and contaminated sediment assessment and management. Todd currently serves as the director of ERDC's Center for Contaminated Sediments, as well as the program manager for the U.S. Army Corps of Engineers Dredging Operations Environmental Research Program. Todd received his BA and Master's in Biology and Zoology from California State University in Fresno, and his PhD in Biological Oceanography from North Carolina State University. And with that, I turn it over to you, Todd.
Thank you, Rula. Good afternoon to everyone. I'm going to talk today about contaminated sediments, largely in the context of two SERDP projects that our large team has been pursuing over the last couple of years. There's a lot to explore in the area of contaminated sediments, given the complexities of remediating, first assessing and then remediating these kinds of projects. But the focus of our research has been on this intersection, if you will, between biology and chemistry that drives exposure at these sites, and then what information you can derive from that expanded understanding to inform how we develop resilient remedies. The agenda, briefly for this 25 minutes, I'm going to talk a little bit, introduce contaminated sediments for those less familiar with the subject area, mention some technology advancements, which I think are offering a considerable amount of additional capability to us, these advancements over the last 10 years roughly. Then highlight some of the results from our work at this intersection of biology and chemistry, and then talk about some of the implications of that work in regard to developing resilient remedies. So contaminated sediments are a widespread problem in the United States. I guess I could add here parenthetically that it's a wide-scale problem around the world. The U.S. is by no means the only country that is struggling with how to manage this problem. Several years ago, more than a decade ago, the EPA did a broad assessment with some uncertainty, but concluded that we had on the order of 1 billion cubic yards of superficial sediment that posed some potential for risk. The U.S. Navy, though it's not the only element within the Department of Defense that has contaminated sediments that it has to manage, but the U.S. Navy probably has the lion's share of these sites. And the costs of these cleanups are considerable. I think the range that I've provided here on this slide of $10 to $100 million per site probably captures 80% of sites, contaminated sediment sites that are ongoing activity within the Superfund program now in the U.S. There are a number of uncertainties concerning these kinds of projects, which to greater and lesser degrees, depending upon site conditions, are driving decisions as well as costs. Bioavailability remains a complicating factor, and its assessment or lack of its assessment is certainly influencing the dynamic of these projects. Risk estimates are intentionally, in fact, biased high. But I think some of the ways that we account for uncertainties that exist now even compound that issue, resulting in very low cleanup levels. In fact, levels in some cases that are so low that they are unattainable, perhaps using traditional remedies. And then we have the uncertainties concerning long-term remedy performance. How are these remedies going to perform over 5, 10, 25-year periods? So two specific problems I'm going to discuss this afternoon is how we improve our exposure characterizations. And the basis of the research we've engaged in on our team for the last several years is, again, the role of biology, in particular functional ecology, how animal physiology as well as animal behavior, life history, the way that interacts with sediment and water, how that influences exposure. There are literally hundreds of benthic species that can inhabit estuarine, for example, and marine habitats, but our characterizations of risks generally only cover a small subset of those species if you reflect upon the types of biological assessments, the types of toxicity tests, for example, that we employ. So our interest is in incorporating this aspect of ecology, that is functional ecology, how the animals interact with the contaminated sediment or the contaminated media, and to use that to improve the way that we characterize risks and design remedies. The other problem concerns the nature of the remedies themselves, and how is it that we strive toward what I've characterized here as a resilient remedy, a remedy that is able to absorb, if you will, an insult, for example, recontamination from external sources, and recover from that with minimal intervention by us. So contaminated sediment sites occur frequently in urban environments, not exclusively, but frequently occur in urban environments where we have many different sources of contaminants in addition to the contaminated sediments that are in place at our site that we're trying to address with a remedy. So the question is, and it's what this figure on this slide is intended to represent, the question is, how do we design remedies which not only acknowledge the fact that there are sources beyond and outside of our site, but also incorporate aspects within the remedy which can in some way deal with those external sources. There are many complications that this complex urban setting bring to bear, not only in regard to risk assessment, but in terms of identifying causes or the allocation process within cleanup sites, and as I just indicated, in designing or incorporating resilience into our remedies. The technology advancements we're going to highlight in this brief seminar, one of which is passive sampling. Over the last 10 years or a bit more than that, actually, a number of approaches and technologies have been developed and to some degree actually been extended and applied at active remediation sites. I've listed some of these here, which emphasize the medium by which passive sampling is being conducted. Essentially, it represents a medium into which and onto which contaminants can absorb, and placing these, for example, bits of plastic, in the case of polyethylene devices, into the environment. Contaminants absorb into that medium, and then we can remove the passive sampler from the sediment or from the water, extract chemically the contaminant from that material, and with some mathematics derive an estimate of the concentration, in the case of polyethylene devices and similar, the concentration of freely dissolved contaminant in that matrix. This is an example from a couple of our team members, Loretta Fernandez and Rob Burgess, where they applied polyethylene devices on the Palos Verdes Shelf off the coast of Los Angeles in California. In the first panel, on panel A, you can see the orange lines represent the concentrations which have been calculated from the passive sampling devices which were deployed in the sediment of the overlying water off the coast over Palos Verdes Shelf. And panel A represents an area of the site, the contaminated site, which had been capped with clean sediments in a demonstration project, and panel B shows an area that did not receive the cap. So you can see the difference in the two profiles here. I would point out in particular that this area here, which is lower concentration in poor water, freely dissolved, than here in this non-capped area at the same depth. But you can see evidence here at the top, at the very superficial layers of sediment, at the very surface of the column, evidence of recontamination, of sediment or contaminants becoming associated with the surface layers after the capping had taken place, illustrating the need of developing mechanisms that can account for or deal with this issue of recontamination. Activated carbon is another technology that has been developed over the last decade or so. And this makes use of one of the important attributes of activated carbon, that being the large amount of surface area represented by activated carbon. 1 gram of activated carbon contains 500 square meters of surface area. It's a tremendous amount because of the microporosity of the material. And for that reason, will strongly absorb, for a number of reasons, will strongly absorb organic contaminants. In this slide shows some of our earliest work, is also supported by SERDP, where we incorporated activated carbon a little over 3% by weight into sediment from Hunters Point in San Francisco. And on this slide here, we looked at the amount of bioaccumulation that occurred in amphipod leptochirus and a polychaete neanthes. They were exposed either to just sediment that was not amended with activated carbon versus sediment that was amended with the activated carbon. You can see that we got as much as, well, it was about 80 to 90% reduction in exposure or bioavailability as a result of incorporating activated carbon into that sediment. So you can see how we might go forward in developing a resilient remedy. One form of this, it's a slide I've adapted from Upal Ghosh, one of our current collaborators and team members, where we can employ passive samplers, and maybe we have some understanding of the biology of animals in a contaminated sediment. We can assess by collecting tissue and deploying passive samplers exposure processes in that case. And then you see in the second case here, where we've applied a cap, let's say, of clean sediment. Into that cap, we've incorporated activated carbon particles as absorbent phase. So that when that cap, for example, becomes recontaminated on its surface from surrounding sediments or from the overlying water column or other sources, this absorbent that's incorporated in the cap can provide a capacity, if you will, to absorb contaminants and manage exposures, not only from recontamination above, but also migration of contaminants through pore water into the cap from below. Okay, so to look at some data from our previous SERDP project, our team's work here, looking at the role of biology, and I'm going to just focus in on a few key points of data here. But we worked with 2 amphipod species that you see on the left there, Eostorus and Leptochirus, a polychaete worm, neanthes, and then a clam, Mercenaria. And you can see in these figures here the difference biology makes in terms of the kinetics, that is the rate and the nature of uptake of PCBs from New Bedford Harbor in this particular case into these different organisms, which have different biology, different physiology, and different behavior with respect to the sediments. And that translates, as you can see in this table here, to differences in terms of the BSAFs, the biota sediment accumulation factors. It's a simple ratio of the lipid normalized concentration of the contaminant in tissue to the organic carbon normalized concentration in sediment. Biology matters, as you can see here, in the case that these BSAFs for the same sediment for different species vary over more than an order of magnitude. So in a series of experiments we set up, which are complicated, we tried to isolate some of the compartments to which animals are exposed in the environment. And you can see a diagram here at the bottom of the slide where we had what we call pathway isolation chambers, overlying water, pore water, and whole sediment. Essentially, these were boxes within boxes. So we had a large aquarium, which you can see pictures of. And in that large aquarium, we had the contaminated sediment that we were using, for example, New Bedford Harbor. Then we had separate glass boxes where we varied the content of those glass boxes. In the case of overlying water and the pore water pathway isolation chambers, we had a fine sand added. And then the whole sediment, it was the same contaminated sediment as was in the aquarium. In the pore water box, one of the panels, glass panels, was actually replaced by a Teflon filter to allow the movement of pore water from the aquarium into the pathway isolation chamber. So here's some data. This is for polyethylene devices in exposures using New Bedford Harbor sediment contaminated with PCBs. And we're only going to look at a subset of this and make a few points. But these polyethylene devices show some obvious things, perhaps, that we're going to see higher pore water concentrations. For example, in the sediment pathway isolation chamber, which is here in the green, the pore water isolation chamber and the overlying water isolation chamber here are pictured in the blue and then the red columns. So we're seeing just a fraction of the contaminant within these, though we are seeing contaminant move across the boundary here. In the case of the overlying water, the only way that the contaminant is getting into that chamber is through the water above the chamber. One of the other things that we did in this particular series of experiments is that we included we introduced novel PCB congeners into the overlying water, in this case showing data for PCB 24. This congener was not present in the New Bedford Harbor sediment itself, so it was only present in the system because we added it. And we did it in this case to be able to evaluate the extent to which overlying water was contributing to exposures in these organisms. And you can see the influence here that these animals living in the sediment to varying degrees are experiencing exposure from PCB 24 in the overlying water, even though it's not associated with the sediment. And here we see bioaccumulation into the tissues. In the first panel, this is bulk tissue concentrations just for PCB 24. That is just the contaminant we added to the overlying water. Bulk sediment tissue in the first panel, and then lipid normalized in the tissues in the second. You can see the difference biology makes here. Ananthes, this polychaete, receives relatively little exposure from overlying water in this system, in contrast to, for example, Leptochirus, which is receiving a considerable amount of exposure to overlying water based upon how, again, it interacts with the sediment itself. Here's bioaccumulation of sediment PCBs from the Bedford Harbor sediment itself, again showing some additional differences across the species here in terms of bulk tissue concentrations, and in this case, in lipid normalized case. We can see differences here. For example, Leptochirus, almost biological sponges for PCB, showing higher bioaccumulation across these different treatments or isolation chambers compared to the other species that we were evaluating. We also looked at a different sediment, bioaccumulation of PCBs from Bremerton in the greater Seattle area. In this case, first I would point out that the relative bioavailability for sediments from Bremerton to these organisms is much higher, in fact, than in the case of New Bedford Harbor, twice the relative bioavailability, in fact. And we can also see differences here related to the species themselves. If we look at the lipid normalized case for, again, the PCB 24, we see relatively little here. And in the case of Bioaugmentation, we see relatively little, whereas before we saw more in the case of the amphipod, because this sandier sediment from Bremerton seemed to allow Eostorus to burrow deeper within the sediment, such that its exposure to overlying water resembled more that of Neanthes in this particular sediment. So across the sediments, the behavior was being affected. And we can also see the influence of PCB congeners on uptake. In this lower panel here, we have BSCF, the biosediment accumulation factor, expressed for different homolog groups of PCBs. And there is a difference. There is an influence of the number of chlorines and the relative bioavailability. And in fact, we see differences across sediments, as you can see reflected here in the case of Mercenaria, a substantially different relative bioavailability between Bremerton and New Bedford for the same congeners. We did some treatment studies in this particular case where we looked at applying a thin, 2-centimeter layer of sand cap over the contaminated material, and we looked at activated carbon mixed in the top 2 centimeters in a series of experiments, and found that we can see, for example, the influence on pore water concentrations using looking here at the polyethylene device concentrations. Where we applied the cap, we see low concentrations in pore water when we provide for this sand cap within the cap itself, at about the 1 centimeter depth level in applying the cap. And but we can still see in some cases, the influence of the overlying water within the cap itself. In terms of bioaccumulation of PCBs in these treatments, we see differences across the animals themselves. We didn't, in this particular case, see any statistical influence of applying activated carbon under the conditions that we did it in this case. It was a relatively low level of mixing for a short period of time. But we do see the influence of this really small cap, 2-centimeter cap, resulting in reduced exposures to Eostorus as well as Leptochirus, but less so in the case of these larger animals, Mercenaria and Neanthes. Again, biology matters. Our next experiment, series of experiments actually is ongoing in our current SERDP project, where we're trying to bring all of this together in what we're calling Quantitative Thermodynamic Exposure Assessment, or QT, where we're combining passive sampling techniques with modeling to provide insight into how to develop resilient remedies for contaminated sediment sites. In particular, focusing on how activated carbon amendment can be used to enhance the resilience of in-place remedies, like capping. This is the experimental design for this particular study, where we're conducting, and we're in the middle of these experiments right now, and we're generating data from these. Similar to the previous case, we're using what's represented here by these blue straws, polyethylene devices. We're having a series of exposures that aren't receiving any external inputs of PCBs, novel PCBs that we're adding, and then another series where we are adding novel PCBs with no animals in experiment 1, with animals added, an amphipod, a polychaete, and a fish. And in this case, where we're incorporating activated carbon directly into the sediment itself, as we had done previously, but to a greater depth, adding a sand cap, and in experiment 5, adding a sand cap into which we've added activated carbon or amended the cap itself. And the final experiment, where we're also going to incorporate activated carbon by introducing it periodically into the overlying water as a way of trying to control the influence of inputs through the water column. This is just some figures showing all the ongoing experiments in the aquarium. You can see, for example, in this slide, the sand cap overlying the contaminated material in this particular aquarium, and the 3 species that we're currently using here. This is Saprinodon, sheepshead minnow. So, conclusions. We're developing evidence, we believe, through this emphasis we're placing upon the intersection between biology and chemistry, which we believe will lead to doing better exposure characterizations for contaminated sediment sites. It'll improve our risk assessments, and I think also improve our decision making with respect to remedies. Passive sampling has grown significantly, I think, in maturity over the last 10 years, and to the point where I believe it's something that should be readily and frequently integrated into RIFSs now in active science. In situ remedies are in fact vulnerable to threats from below and threats from above. If you have contaminated sediments that you're leaving in the environment, that sediment can influence the remedy, for example, a cap. But more particularly of concern to us in this particular series of experiments is how external insults, contaminants from off-site or beyond our control, are going to influence the long-term performance of these remedies, and how can we incorporate aspects into these remedies which will add resilience, ability for these remedies to, if you will, absorb that insult and continue to perform. And I think incorporating that particular objective into our design will improve the effectiveness of our overall remediation program. So with that, I'll conclude, and I guess we have a short period for questions.
Yes, thank you so much for a very informative presentation. I would like to remind our audience to please submit questions by using the chat box on the left-hand portion of the screen. Also, for additional information about this project, you could refer to the SERDP and ESTCP website. Dr. Bridges, we have received a number of questions that I'll be posing to you. For starters, how large a role do biological processes play in exposure in comparison to chemical processes?
That's a good question, a very broad question, and like many answers from scientists, I guess I would say it would depend. And it will depend on the conditions, at the site itself. I mean, I think the data that I've shown this afternoon illustrate that biology does have a role. And in fact, the role of biology will also vary depending upon the sediment. As animals behave differently, the same species can behave differently and interact with that sediment in different ways depending upon the characteristics of that sediment itself. I think in general, over the last, say, 25 years of work in the area of contaminated sediments, I think the majority of the emphasis that perhaps has been placed on trying to understand the role of chemistry in driving exposure and thermodynamics in regard to that. Less emphasized, but I think important for us to resolve some of the remaining uncertainties is the biology, and in particular the intersection between biology and chemistry. So I guess I would say, it depends. It's something we need to factor more into our risk assessment. The animals, the hundreds of species that might exist within the sediment bed at our sites are not just a bag of fat. They have behavior, those species, and they have physiology, which is going to influence their exposure. And if we don't account for that to some degree in our risk assessments and in our remedy designs for that matter, I suspect we're going to be surprised, maybe in unpleasant ways.
Thank you, Todd. Another question, have you given some thought on how to scale up your lab-based experiments into a real-world field application?
That's a good question, and scale-up is a significant issue in many cases. I mean, we did follow the first work that Dick Luthi led, where we were using activated carbon laboratory with an ESTCP project that was conducted at the Hunters Point site. And there were a number of papers that were published as a result of that at the scale of, sites that were the size of your living room, for example, or your bedroom. But then there is going to the next level of sites that might, include an acre or a hectare. And there could be different scale-up issues for moving across that whole spectrum of size. I mean, personally, I think one of the ways that we can advance our practice at real contaminated sediment sites is for us to make more use of what I would call demonstration projects or pilot projects earlier in the process of investigating and moving through a feasibility study. There are just limits to what we're going to be able to resolve in form of uncertainty about remedy performance from laboratory studies or modeling studies. I think there are going to be variations that are occurring in the field at different project sites that can only be addressed by piloting and demonstrating different technologies that we can move forward with in the case of those that are performing well in those demonstrations.
Thank you very much. Our third question, do the sand caps influence the number and health of sediment-dwelling organisms, since it is certainly a change in their environment?
Right, that's a very good question, and it will, of course. If you're putting a sand cap on top of what previously was a fine-grained sediment environment, you will change the species composition in that community. Now, if you take maybe a longer-term view, you may have placed a sand cap on that site, but if the sediment transport processes in that environment have resulted in a fine-grained sediment environment, what you might find over some period of time is that sand cap is going to be progressively buried and will become finer over time. And actually, as a consequence of that, you'll maybe see the benthic community shift over that period of time in response to those changes. I think one of the benefits that capping does offer is our ability to incorporate habitat objectives into the cap design itself. Sand caps certainly present some conveniences in terms of construction, but there's no reason why one can't incorporate a finer grained material into the surface layers of a cap design. For example, if you have a desire to incorporate a habitat, if you will, layer into a cap.
Great, thank you. Another question for you. You mentioned risk assessments earlier. What do you think are the key factors for improving risk assessment practice for contaminated sediments?
Well, I think the data that I've shown illustrates some of the things that we can do. First is recognizing that, for example, taking BSAF values out of the literature and trying to apply them, for example, to your site may not be an advisable thing to do, given the amount of variation across sediments that one can observe, depending upon, for example, the nature of the carbon present at a site. You may have one site that has a lot of black carbon in it, which is going to give you a much different BSAF, a much lower BSAF than a site which has relatively little black carbon. But I think the other, I guess one of our major messages is that we need to be maybe paying, I believe, more attention to the interspecific, the differences that exist among species. And if we're basing our exposure estimates in this critical transition, if you will, from sediment concentration to benthic tissue concentration, if we're basing that assessment on a very limited number of species, we may very well be introducing, and probably are introducing, bias into our risk assessment. The nature of that bias might not be clearly understood unless you have a broader understanding of the species diversity and how much exposure may vary among those species. And it also influences, I believe, remedy design and factors that we should be considering in terms of a cap design with respect to species behavior.
Thank you so much. One last question. How much more costly will it be to incorporate resilience into remedies, broadly speaking?
Well, I mean, there's published work that has tried to estimate, for example, based on the costs of carbon that one can look at. I would choose to kind of reframe the question a little bit and think about the additional costs of developing a more resilient remedy versus redoing a remedy or somehow addressing a weakness in a remedy when you get to a 5-year, for example, a review period based on monitoring data. I think it's something that we need to give much more attention to. We may end up having to invest a little bit more in terms of material or time to develop what I'm characterizing here as a resilient remedy initially. But the payoff for that is that the remedy is going to perform more reliably and consistently over a longer period of time in cases where these sort of external influences of ongoing contamination, which are beyond our reach to control, have a potential to influence that site and the remedy over time. So I think it's, in some cases, maybe it's pay me now or pay me a lot more later. And I think we need to be viewing remedies at contaminated sites, particularly in situ remedies, as a kind of a dynamic process. I think there are a relatively limited number of cases where we're going to take a single action and walk away forever, in particular in cases where we have in urban environments these ongoing sources of contamination.
Thank you again for a wonderful presentation and for answering some of the questions that we've received. And it is now time to turn this over to Dr. Kevin Sowers, who is Associate Director at the Institute of Marine and Environmental Technology and Professor at University of Maryland in Baltimore County. Dr. Sowers' research over the past 20 years has focused on the process of microbial dechlorination of PCBs and the development of sustainable in-situ technologies for treating PCB contaminated sites. Kevin has been a principal investigator on several DoD, NSF, and NIEHS grants focusing on identifying PCB dechlorinating bacteria, characterizing their physiology, and developing approaches for scale-up and deployment of both anaerobic dechlorinators and aerobic degraders at PCB-impacted sediment sites. Dr. Sowers is currently conducting field trials in collaboration with federal, municipal, and private stakeholders. He received his doctoral degree in anaerobic microbiology from Virginia Tech in '84 and conducted postgraduate research at the UCLA Department of Microbiology and Molecular Biology. And with that, I'll turn it over to you, Kevin.
Okay, thank you, Rula. As my introduction indicated, we've been working a number of years in understanding the process of PCB degradation. The work began with trying to understand the anaerobic processes responsible for dechlorinating PCBs, because at the time there was very little known. It was just known that the process occurred in sediments, but we didn't know how it occurred. So much of the early work was trying to identify these organisms, learn a little bit about their physiology, and finally then our ultimate goal was to go back into the environment and see if we could take that knowledge and accelerate the process of natural attenuation in the field using in situ bioremediation to treat PCBs, and that's where we are now as a result of ESTCP funding. The project team includes myself, Upal Ghosh with Sediment Solutions, and I'll talk about his connection with this in a moment, but largely it's through his company Sediment Solutions and his product SediMite. And then Harold May, who is also an anaerobic microbiologist at the Medical University of South Carolina. So the three of us are collaborating on this project, this current ESTCP funded project. So my agenda will include some background on the process we're using, a description of the technology. Then I'll discuss some treatability studies we've done, and then finally I'll have some preliminary results from our site demonstration at the Quantico Marine Base, and finally I'll summarize those results. So just to mention PCBs, it's a biphenyl structure with varying amount of chlorination, halogenation of that structure. And depending on the position of the chlorine, you have 209 potential congeners that can result depending on the position and number of chlorines. This was considered a miracle chemical at one time because of its great chemical stability. It's stable against oxidation, and it's stable at high temperatures. So it had a range of applications since it was first produced in the 1930s. The compounds are hydrophobic, therefore they tend to bioaccumulate in lipids, particularly in the fatty tissues of larger mammals, aquatic mammals, and they are a toxicity concern. Not all PCB congeners are necessarily that toxic, but depending on the congeners, some have dioxin-like structure and characteristics, some can be teratogenic, some can be potential causes of cancer. So there is a toxicity concern with having PCBs in general in the environment. And finally, the sediments are the ultimate global sinks. PCBs are hydrophobic. They tend to absorb to organic material. It settles into the sediments. And as a result, those sediments flow out through waterways, finally out to the oceans. And it's the sediments themselves that tend to be the sinks for PCBs. So we're not talking so much about a groundwater issue, but a sediment issue. So the problem statement, many sediment sites, particularly many DoD sites across the country, are impacted with PCBs. And the current technologies are either destructive to the environment or may not sufficiently reduce the risk. The two common technologies now, the most common is dredging, often with post-treatment or landfill, and also capping. And each has its advantages and disadvantages. What we're trying to do is develop an in situ treatment that's a bit more passive, and we combine our treatment with bioaugmented activated carbon. The advantage of this we see, if we're successful, would be first, it's very cost effective. It's essentially the cost of doing a thin layer cap. It both sequesters and degrades the PCBs. So the activated carbon immediately sequesters the PCBs over the short term and keeps them out of the food chain. And on a slightly longer term, then the microorganisms that we apply to that then break down the PCBs. The method is rapidly deployed, and the site we're working at now, the Marine base, we're working at an active site, so being able to very rapidly deploy and get out of the site as soon as possible is an advantage. It's also minimally invasive. We require very little in the way of large equipment. It reduces the overall health risks associated with sediment disruption, which is commonly associated with dredging. Is reduced overall energy risk, energy use, and it negates the requirement for extensive waste management and habitat restoration. And finally, in the future, we hope that this will be the approach we're taking will be potentially applicable to other contaminants as well. So how the process works. The degradation of PCBs occurs by two processes in the environment. The first is anaerobic dechlorination. These are respiratory anaerobes. They actually use PCBs as terminal electron acceptors. And in doing so, they displace the chlorine on the carbon group is reduced with a hydrogen. And this process is generally will attack higher chlorinated PCBs. Generally, the process slows down or stops after there are no longer any chlorines with flanking chlorines. And at that point, as I said, the process slows down or stops. Aerobic degradation, on the other hand, will not attack higher chlorinated compounds, but will only attack the lesser chlorinated congeners once the 2, 3, and 3, 4 positions of the rings are opened up as a result of anaerobic dechlorination. So the combined processes working together result in the complete degradation of PCBs. And since many of the PCBs are higher chlorinated compounds, this is 1254, Aerochlor 1260, both the anaerobic and the aerobic processes are necessary to get complete degradation. So just a general description of the approach we take. We first, the first thing we do is we establish mesocosms. These are 2-liter glass mesocosms that we run in the lab, and I'll describe those briefly in a moment. But essentially, we try to simulate the conditions at the site, and we test sediments and water from the site, do different types of amendments, and then we find out which combination of amendments are best applicable to that particular site. Next, we then scale up the microorganisms. Depending on how many organisms and which organisms we need, we then scale them up for that particular site. It's then deployed. We actually deploy the microorganisms on a product called SediMite. I'll describe that in a moment, but it's agglomerate of activated carbon, sand, and clay that's been developed by Sediment Solutions. And we essentially piggyback on that technology to get our microorganisms down into the sediment. The microorganisms on the carbon particles are then naturally dispersed as a result of bioturbation. And it's this natural process then that moves our organisms from the surface down throughout the benthic zone of the sediment. And finally, we then assess the fate of the PCBs by a number of ways: gas chromatography to look at the fate of the PCBs, and then we use molecular approaches to determine the fate of our microorganisms. Are the numbers staying the same, going up? Are they still there a year later, and so forth? So the site we've selected for the ESTP study is Abrams Creek in Quantico, Virginia. This is on the Quantico Marine Base. It's an 8-acre watershed outflow to the Chappawampsic Creek. The PCB concentrations range from 3 to 5 milligrams per kilogram. Generally, all the samples we've taken fall within that range, so it's rather homogeneous. And we think the possible source of the PCBs, and it's not certain, may have been from pesticides. At one time, PCBs were used as extenders for pesticides to enable them to be retained longer on vegetation and so forth. And this is one possible source. And 1254 and 1260, Aerochloris 1254 and 1260 were commonly used for this reason. The water depth is about 100 to 250 centimeters. So we have a meter or so of water column to go through. The organic carbon is about 28,000 milligrams per kilogram, so we have a good source of carbon to feed microorganisms. And finally, we have road access. So it's a very accessible site in terms of actually deploying the treatment. So the treatability studies I mentioned, they're conducted in 2-liter recirculating mesocosms. The sediment samples are taken from the site. We take all the samples, we homogenize them so they all have the same concentration of PCB and they're all homogeneous in terms of other conditions. We also take water from the site. The sediment is then put into the tanks, and we use the water from the site. And each tank here, it's run on a multi-channel pump. What we do is we circulate the water from the tank to a flask. There it's resparged with air so that we can maintain the same oxygen concentration in the water column that you'd find at the site. We don't want these to become stagnant and go all anaerobic. The water is then pumped back up into the tank, and then it goes through a linear manifold, and there's a slow flow rate over the surface. So it doesn't perturb the sediments, but it's enough to keep the water column oxygenated at a consistent oxygen concentration. And we run these mesocosms usually for a period of 4 months to try and get an idea of which treatments are best. And so we'll do a variety of treatments with, in this case, we did different types of organisms, different numbers of organisms, additional carbon sources, to try and find out the best condition. And then the PCBs in these mesocosms were then monitored for 120 days. So this slide shows the effects of treatments on the PCB homologs. Down below on the y-axis, I have the homolog groups, 1 chlorine, 2 chlorine, 3 chlorine, and so forth, and then the concentration of PCBs. This is a mesocosm that was treated with SediMite alone. No microorganisms were added. And as you can see, there is no significant difference, and there's no statistically significant difference between the homolog groups after 0, 30, 60, and 120 days. On the other hand, this mesocosm, which was treated with the anaerobe, in this case we used a halorespire known as dihalobium chlorochlorosea, and an aerobe, which was, the strain name is LB400, but this is a PCB degrader. Use them together, and we put concentrations of 10 to the 5th cells per gram of sediment. And as you can see, after a period of time, you see a reduction in all of the homolog groups. And this is significant because this means that not only do we have evidence of anaerobic dechlorination of the higher chlorinated congeners, but we're also seeing aerobic degradation of the lesser chlorinated congeners. So both groups are working concurrently. So the abiotic, there's no degradation. We add the SediMite with the bioamendment, and we get 72% degradation was observed in 120 days. And as I mentioned before, we see a decrease for all the homolog groups, meaning both organisms are working together. When we look at 3 of these mesocosms, this was looking at the different concentrations of cells, 10 to the 3 cells per gram, 10 to the 4th, and 10 to the 5th. The results show that 10 to the 5th cells are most effective. That's where we got the most effective treatment of the PCBs after 120 days. 10 to the 4 cells per gram, less so. 10 to the 3 cells per gram, we see some change, but it's not statistically significant. There was a slight change. Whereas in the controls not shown here with their abiotic, we see absolutely no change at all. So the number of cells per gram has a significant impact on the rates and the final concentration of PCBs at the end, going from no significant change, 50% change, and 72% change. So for our study, ideally we want to look at the fastest rates. We'll be using 10 to the 5th cells for the site. When we look at the treatment with depth, this graph here shows the homolog groups. This is the upper layer here, so this is the top half of the sediment column, and this is the lower half of the sediment column. And what we see is we see the same amount of degradation for all the homolog groups in both the top and the bottom. And when you look at the mesocosm, we see this active worm activity. So there was ample mixing as a result of the worms, and the worms were from the site, that resulted in mixing of our organisms throughout the sediment column. So this indicated that the active benthic activity actually served us in terms of distributing the organisms and enable them to be distributed throughout the sediment column. This slide shows the scale-up. Once we had determined that we would need 10 to the 5th cells per gram, this would be for cubic centimeter on the surface of the sediment at the site, we were going to treat approximately 1/4 of an acre. We would need 10 to the 12 cells total for this particular site in order to do our treatment. We've developed technologies. I'm not going to get into details, but essentially we were able to scale up our anaerobic halorespirers. Required a 200-liter reactor, but we were able to obtain 10 to the 12 cells. The organisms are scaled up in the reactor. They're then harvested with a continuous centrifuge shown in the back here. And then the concentrated cells are transported to the site in a stainless steel, 20-liter stainless steel Cornelius flask. And those were sealed under nitrogen. For the aerobes, we could get 10 to the 13th organisms with just a 20-liter fermenter, and this was harvested directly into a Cornelius flask for transport. Of course, we needed to know how long these cells were stable before we got to the site, so we know how far ahead of time to grow these organisms. And this slide shows the cells after we harvested them and put them into the Cornelius flask. We had 10 to the 9th cells per mil transferred into the Cornelius flask. These were concentrated cultures. We stored them at just room temperature. And we looked at both the total number of cells, this is the aerobe, and the total and the viability of those cells. So we know that they were viable as well for both the aerobe and the anaerobe. And we looked at the total count and viability after immediately after harvesting, 1 day later, 14 days later, and 30 days later. And what we found is as long as we used the cells by 14 days, we still had nearly 10 to the 9th cells. And for this particular case, this was more than enough cells that we needed for the site. Even after 30 days, the aerobe, we still had almost 1 to the 9th cells, which would be needed for the site after 30 days of storage. And we lost about half of them, of the anaerobes at that time. But still, even at that time, if we kept them for 30 days, we still have ample number of cells to do our test. In the case of the site I'm going to talk about today, they were used within 14 days, just to be on the safe side. Now, SediMite. SediMite is a product that was developed by Upal Ghosh and collaborators. They have a startup company called Sediment Solutions. And it's essentially a agglomerate of activated carbon, a weighing agent, which is sand, and an inert binder. And these form pellets, and these pellets will go through the water column. The problem with activated carbon, if you just put it on the surface, most of it would just float. So what this does is this gets the activated carbon down into the sediment through the water column in the sediment. And with earlier SERDP research, we were able to show that this activated carbon didn't inhibit our organisms. One of the questions was, does bioavailability, reduction of bioavailability to benthic organism also affect microorganisms? And we found that adding activated carbon to our microorganisms with PCBs did not adversely affect their ability to dechlorinate and degrade the PCBs. So stepping off the SERDP project, we got into the ESTCP project, and we developed a method for using this as a carrier for our microorganisms. We essentially spray our organisms onto the SediMite, and then we disperse the SediMite using techniques that have already been developed by Sediment Solutions. And the advantage of this is that using the activated carbon immediately reduces the viability to benthic organisms in the sediment. Well, and it also provides a solid substrate for biofilm formation by our microorganisms. So the advantage of this is we not only remove the PCBs, reduce the bioavailability of the PCBs as a result of the activated carbon, but our microorganisms then degrade those PCBs. And SediMite's currently licensed Sediment Solution, and that is being transitioned to the field now as a in situ treatment. So one of the questions, of course, was, will our organisms stay absorbed to these pellets as they go through a water column? So we ran an experiment where we sprayed our organisms on to the pellets and then passed the pellets through a 1 meter column, collected the pellets, and then we did both a total cell count and a viability assay. And the bottom line here was there was no significant change between the number and viability of the cells on the pellet when we first spray them and after they pass through the water column. So that indicates to us that the organisms will be carried through the water column and will have the same titer once they get into the sediment. So for our test design at the Marine Base, Abrams Creek, we outlined 4 plots. We have 4 10 by 4 meter plots. The first plot is completely untreated, and the second will be SediMite alone with no microorganisms, and the third and fourth would be the bioamended SediMites. These would be duplicate plots. Within each plot, there are 5 subplots, and within each of the 5 subplots, there's a random grid. So we randomly select samples, 1 sample from each of the plots. And then we have an exterior zone, which we won't sample, and that'll account for any drift or for any irregularity in the plot, the footprint of the SediMite once it was put into the plot. Plots were sampled 1 month before treatment, 4 months after treatment, and then we'll be sampling again 12 months after treatment. As I mentioned before, each of the plots is randomly sampled within the 5 locations, so we have 5 cores during each sample for each plot. And then the cores are divided into sections of 0 to 7.5 centimeters, which is the main benthic zone, 7.5 to 15 centimeters, and finally 15 to 30 centimeters. So 0 to 3 inches, 3 to 6 inches, and 6 to 12 inches. And then each of those subcores are then homogenized and tested for PCBs. So the deployment, we went to the site on April 27th, so this is all still relatively new. We brought 6,000 pounds of SediMite. That was enough to do the 3 plots. We had a staging area where the microorganisms were inoculated onto the SediMite, and here we're mixing the organisms, the anaerobe and the aerobe, put into a pump, and with the pump and sprayer, we then sprayed the organisms onto the SediMite. We sprayed a known amount of organisms onto a known amount of SediMite, so we knew the exact concentration of cells per gram of SediMite that would end up in the sediment. Once they were sprayed, they were transported to the water side in 5-gallon buckets, loaded onto a boat, and they're contained here. These are the bioamended SediMite in these 2 bins. And then they were then deployed using an air horn. Now, we tested a number of methods, but the air horn seemed to be the best for this particular case. The air horn is a air ventura. It's run by a high volume compressor shown here on the shoreline. And what happens is, as a result of the suction from the air in the ventura, the pellets are pulled up from the bins, and then they were deployed then in each of the 5 subplots. So we do 1 subplot at a time. And our goal was to get 1 centimeter layer of 1 cubic centimeter layer of SediMite with microorganisms on the sediment. Deployed passive samplers. So at the time, we did is we deployed passive samplers using polyethylene membrane. These membranes were put into stainless steel screened frames. The frames were then inserted. We developed a rod that would enable us to insert these down to the surface sediment. And the way they're arranged in there is we have a sampler down in the sediment itself. This will be sectioned off into the top 3 inches and the lower 3 inches when we do the assessment. And we also have a membrane up in the water column. So we'll want to determine the effect of the treatment not only on the total PCB concentrations in the sediment, but we want to see what the effect is on the aqueous concentration of PCBs, both in the pore water and up in the water column. So we'll be monitoring that as well. We've done our first sampling. This was 140 days after treatment. This is out on the offshore sampling using a coring device. The cores were then brought to the lab. They were chilled on a cooler, brought into the lab, and they were subsectioned. And you can see some of the cores here. We took the first 0 to 7.5 centimeters, 7.5 to 15, and then finally the bottom area of the core, which we don't expect to see any effect there simply because this treatment's only going to be effect up in that benthic region. So 20 cores were collected, 5 for each plot. We also collected, we will be collecting next week, the passive samplers. They were deployed at that time. They've been out there now for approximately 45 days, and we'll be collecting them next week to get the data on the pore water concentration and the column in the column. And then analyses on those are pending. But just this week, we did get the initial results from the core samples, and that's shown in the next slide here. This is very preliminary, but what we found, we were very excited about the results. Plot 1, which was untreated, we saw essentially no change in the PCB concentration. Plot 2, which was only SediMite, no microorganisms, again, we saw no significant change in the PCBs in the sediment. On the other hand, in plots 3 and 4, we saw a significant change in PCB concentration after only 140 days. In plot 3, we've already seen 24% reduction in the PCB level. And in plot 4, we already see 36% reduction in the PCB level. And we'll be continuing to monitor this. We'll be going back next year in the spring, and then we'll be reassessing. And as I said before, we don't have the data from the passive samplers yet, but we'll be looking at the passive samplers to see what the different treatment effects are, particularly the amended treatments are, on the bioavailable or aqueous portion of the PCBs. So in summary, PCB levels in mesocosms, we showed a reduction in 72% after only 120 days. We've continued to monitor these mesocosms, by the way, and we've seen the reduction is now approaching about 78% reduction. The methods, we developed methods successfully for the scale-up and the deployment of the bioamendments into the sediment. These microorganisms are scaled up without PCBs, so when we put them back into the sediments, there are no residual PCBs or POP associated with the cells when we put them back. We were successful in deploying the SediMite with the microorganisms in May of this year. And PCB levels, as I mentioned, we see significant reduction after only the first 140 days. We'll be continuing to monitor this. So the next step now is to reassess after a year and then identify the parameters necessary for an effective full-scale treatment at that time. So the benefits of this approach compared to other approaches with the bioamended SediMite, the cost is approximate, but the cost of a thin layer cap, it's approximately the cost of capping in the $150,000, $170,000 range, but it's much less expensive than dredging. And we're not advocating that this is a replacement for any technology, but rather we see it as a complementation. There are certain places you're going to dredge if there are hot spots. You may want to then treat with bioaugmentation after you do the dredging to clean up any residual PCBs. There may be sensitive areas, and if you see here, the risks are low because we're not disturbing the sediments like capping. You're covering them up as opposed to dredging. There's always a risk as a result of dredging up the sediments. So there's a much lower environmental disruption. This would be an approach you might want to use in areas that are environmentally sensitive, certain wetlands, where you don't want to go in and dredge and you don't want to cover it up. You could actually use the bioamended SediMite as an approach. Very low energy use compared to dredging. It's as far as long-term waste management, there's nothing more to do. You put it in place and you monitor occasionally. Habitat restoration, there's no restoration cost required. You essentially treat and walk away. And it's well suited to wetland environments, as I said, where you have environmentally sensitive areas. And with that, I'd be happy to take questions.
Thank you so much for a very interesting presentation. We have received a number of questions. The first one from the Washington State Department of Ecology. And the question is, PCBs may also be associated with PAHs. Would your technology also be able to treat PAHs and other sediment contaminants?
Our goal ultimately is to look at other compounds. For what we're doing, using these particular microorganisms, these are specific for PCBs. But our ultimate goal would be then to use the same approach, but use microorganisms that are specifically degrade, say, DDT, PAHs, other types of contaminants. We'd have to use the types of microorganisms that are specific for those contaminants, but ultimately our goal is to use a similar approach, and yes, try and go after other contaminants as well, and in some cases we know more often than not, it's a case of co-contaminants. So try and treat different contaminants simultaneously. That would be our goal.
Great, thank you. In the Abrams Creek treatability study, how were stream dynamics considered relative to the distribution or redistribution and shifting of sediment during periods of high flow during storms and floods?
Are you referring specifically to this study? Is it?
The question is related to the Abrams Creek treatability study, yes.
We have, there is a flow level here, particularly in the spring after the runoff. And then generally during the summer, that flow slows down. We don't have the results yet, but we did take cores downstream of the plots to try and determine if there's been any shift in the carbon and the microorganisms. And we'll be analyzing those cores both for black carbon content and for our organisms using, we have a very specific molecular assay that enables us to look at the numbers of our specific organisms. So we'll be assessing the downstream area of the sediment. We will be taking that into account. Right now I don't have an answer, but that's one of the things we will be checking for.
Thank you. Was baseline benthic community health collected or evaluated for comparison after treatment?
No, it was not. We did not do any benthic assessment. More anecdotal, we did the mesocosm studies and there was very active benthic activity, but I can't quantify that. It's just anecdotal. The worms were very happy in there. And they stayed alive for about 120 days. Then we started to see the benthic community die off. But those were closed systems. There could be a number of reasons for that, such as the carbon sources they used had been used up. So I don't have an answer. It's not something that we included in our assays.
Thank you. Would this technology be expected to work as effectively in an intertidal area?
In an intertidal area with movement, we have not conducted studies specifically using activated carbon or SediMite. But Paul Ghosh and Sediment Solutions, they've done more of these studies. The risk, of course, is movement of the carbon before it's fully integrated into the sediment. And of course, if the sediment is disturbed in the intertidal regions, then you could expect movement as well. obviously we're going for the low-hanging fruit here. We want to see if it works, and we picked an area that wasn't subject to the tidal changes. I think that would be something that we'll want to address in the future to see if it is effective. It does enough carbon get in there, and is it stable enough over the long term that the organisms can break down the PCBs adequately. I think it's something we need to look at in the future. I don't have an answer right now.
Thank you. What about the use of this technology at sites with higher concentrations of PCBs, up to 100 milligrams per kilogram?
We currently have a site, as a matter of fact, in Alta Vista, Virginia. It's an old sewage treatment pond that isn't in use anymore. And we're looking at concentrations of about 1,000 parts per million, 1,000 milligrams per kilogram. And the treatment has been very successful there. We did a small caisson study, essentially took an oil barrel and took the ends off. And we did a treatment there, and we obtained 80% reduction of the PCBs. And we were going from, in that case, one caisson, it went for about 300 parts per million down to below 50 parts per million. As a result of that study, we've met our goal. We now have large, the 10-foot wide caissons in place to do a much larger study based on that data. And that's ongoing right now. But yes, in that case, we were looking at ranges between 500 and 1,400 parts per million, and they were effective.
Thank you. A question related to the mechanisms of action. Is the mechanism of action related mostly to the sequestering effects bringing PCBs to the Sedimite surface for biodegradation, or is it due to the migration of the microorganisms from the sediment or from the Sedimite into the sediment layer?
We don't know that yet. As a matter of fact, we were just awarded Birta Gellerup is the PI on a SERDP award to look at this on a microscopic level and determine exactly how the microorganisms are interacting with the PCBs. We hypothesize that the microorganisms are forming a biofilm on the particles, and essentially the particles, the activated carbon, are bringing the PCBs to the microorganisms. The Sedimite particles, I mean the Sedimite in there is essentially a powder, so we're talking very small particles when it's distributed. And what we think is happening is the organisms are absorbed to the particles, the PCBs come to the particles, and within a small area you have a very high concentration of PCBs. At that point the microorganisms act on them. That's a hypothesis. We don't know that. But this new study, which is supposed to begin shortly, will be addressing that issue.
Great, thank you. And one last question before we bring Dr. Bridges back into this discussion. Assuming conservation of mass, where does the chlorine go?
The chlorine presumably just goes into the water phase. But you're talking very, very low levels.
Okay, excellent. All right, well, we have one question for Dr. Bridges, and then a couple of questions that we would like you, Kevin, and also Dr. Bridges to opine on. For Dr. Bridges, we heard in a previous seminar in November of 2014 about the importance of grain size on contaminant partitioning. Was grain size of the sediments and sand an influencing factor in your experiments?
Well, I noted there were grain size differences, for example, between the New Bedford Harbor sediment and the Bremerton Harbor sediment. The Bremerton sediment having a coarser quality to it, which I noted today, it seems to have been an influencing factor in shaping the behavior of the animals and thus their exposure. I think the relationship between grain size and organic carbon content, of course, which are closely associated with it, and the potential, not just the potential, but for the particles to have, as Kevin was noting, biofilms on them. That's certainly related to bioavailability overall. The more surface area that you have coated with carbon or containing carbon, the greater the potential absorption to the particles. So there is a complex physical, chemical side to bioavailability. Our work is a little bit more focused on when you introduce biology, a third, if you will, factor into that, it gets even potentially more complicated. But I think that we're, slowly but surely as science rolls forward, being able to resolve some of these sources of uncertainty to improve our ability to characterize exposure, which is which is what we're doing, what we're trying to do, advance our ability to improve the accuracy of our exposure estimates for benthic organisms as well as fish.
Great, thank you, Todd. And then a question for both you and Kevin, starting with you perhaps. What do you see as the future trajectory for the remediation of contaminated sediments?
Well, just as Kevin noted earlier, he referred and made comparisons to, say, dredging. And I think if you look back, say, 20 years ago, or maybe even more recently than that, I think it's fair to say that the default remedy for many contaminated sediment sites was dredging, was a removal action, and that the majority of the investment in that remedy was dredging. And what we have, I think, progressively found through time is that dredging and removal may provide some value and importance, but that there are limits to how much one can reduce risks, because dredging is far less than a surgical procedure. And so I think what we have seen over the last 10 or 20 years certainly is more focus on what we would call in situ remedies, of taking action to reduce risks in place. My expectation would be that that trend will continue, and that there are a number of reasons why I think that's smart. I think the technology is rapidly developing. Kevin's work certainly shows another aspect of that development, to be able to address risks in situ. And I think it's also consistent with the idea of, if you will, adaptively managing these sites, which I think, because of the nature of them, particularly complicated harbor, urban sites, are going to be adaptively managed. We're not going to probably be able to be fully successful in implementing one grand remedy at one point in time and walking away from that and being fully satisfied with the outcome. So I think it's reasonable to start with, as I said, these demonstration projects and pilots, which you progressively scale up, depending upon which ones are proving to be useful and beneficial, and adaptively managing your site through that progression.
Great,
yeah, I would agree with that assessment. I think the more approaches we have available to us, bigger the toolbox, we can adapt the best approach for a particular situation, because every site's going to be different. There may not be one approach that's appropriate for that single site. It may be multiple approaches. As I mentioned before, dredging might be necessary if you have a hot spot or a source point. Well, you need to stop that before you do any treatment, because the treatment isn't going to be effective then. So you're going to dredge that, mediate that, and then use a second treatment, maybe a third type of treatment for a particular site, for treating hotter areas and the less hot areas. The other trend I see is the assessment. There's been a lot of emphasis on total levels of PCBs and sediments. And I think the thought is changing. It's not a true assessment of risk. And it's unreasonable to think some of the numbers out there, some of the levels that are expected, are just not going to be attainable. The other hand, the risk itself is more important. What is the risk to wildlife and humans? And it may be if the PCBs may remain, but if you make them less bioavailable and they're no longer a risk, well, maybe a higher level in sediments is acceptable then. So I think another changing trend is, rather than looking at total PCBs, moving more towards actual risk associated with the particular PCBs at a site.
Great, thank you both for excellent presentations. The next webinar is on Thursday, November 12th, and will focus on the munitions response program on the land-based program closeout. This webinar will feature 3 presenters: David Wright from CH2M Hill, John Jackson from the U.S. Army Corps of Engineers in Sacramento, and Doug Maddox from EPA. Before we conclude, I would like to remind you all that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it if you can please take a moment to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you all for your participation.